Bismillahirrahmanirrahim. Assalamu alaikum, dear class. I hope all of you are fine and safe at your home. In this lecture, first we will study about the microarrays, a brief introduction of the microarrays, then types of the microarrays, then our focus will be on DNA microarray, principle and working of DNA microarray. Then we will study about the types of the DNA microarray and what are the advantages and disadvantages of DNA microarray. Microarrays. As you have performed various kinds of experiments in lab during different courses, and you have seen that you perform experiments in the test tubes. Like for example, we take a simple example of DNA isolation, right? You need certain Eppendorf tubes for isolating the DNA, and you are performing certain identification tests in which you are using the test tube. And these are the large tube, and you need a large amount of the samples to identify a particular compound. So if you think for a minute that if no test tube needed to conduct an experiment, and you can perform hundreds, thousands, or even millions of the individual experiments in parallel with very few reagents. So this is a technique. Microarray technique is a technique in which hundreds, thousands, or even millions of individual experiments they are conducted in parallel, and with the use of very few reagents. And this technique of microarray, it combines the genomics. Genomics means if we want to study all the genes which are present in the genome with experiments, and eventually it is also used for the diagnostic purpose. So thousands of spotted samples which are present on this small chip, and these are immobilized on a solid support. Like you can see, there are different kinds of samples that can be detected or that can be used for the microarray experiment. Like microarrays can be used for the detection of peptides, for proteins, DNA, for lipids, carbohydrate, and small molecules. So we can say that microarray technology is a kind of technology which is used for the experiments which we perform by using different biomolecules as well as the small molecule, because all of these molecules which are the DNA, protein, lipids, and carbohydrates, these are the biomolecules. And we can also use the other small molecules like any antigen or antibody in the experiment. So an experiment with a single DNA chip, for example, it can provide information on thousands of the genes simultaneously. And here you can see the different colors. These are colors are given to the samples by labeling the certain samples with the help of which we identify thousands of the genes. If we talk about that, the sample is of the DNA, thousands of a gene in a single chip simultaneously. On this chip, there is an orderly arrangement of the probe on the support system, which is a solid support. We will see what kind of solid support system we can use and how can we perform the microarray experiment. Now comes the types of the microarray. Depending upon the kind of immobilized sample which is used to construct the arrays and the information fetched, the microarray experiment, they are categorized into different types depending upon the molecule which we are going to immobilize on the solid support. Suppose this is a microchip on which we have solidified or we have attached the different molecules. So these include the DNA microarrays when we are dealing with the DNA, and this DNA could be the small sized DNA molecules, which we will call them as the oligonucleotides, or we are using the cDNA microarrays. And this microarray is also known as a kind of the expression analysis. We can call it as the expression analysis. In this experimental setup, cDNA, which is derived from the messenger RNA of known gene, it is immobilized on the solid support, like these red dot or blue dot. These are the cDNA molecule. The sample that we are going to perform, it has the genes from both the normal as well as the diseased tissue. Spots with more intensity are obtained for diseased tissue genes if the gene is showing the overexpression pattern in comparison to the genes in the normal person, which are responsible for the normal genes which are responsible for the disease. So we can use the DNA microarray for expression analysis, as well as it is also used for mutation analysis. For using this for the mutation analysis, there is a need to use the guide DNA, which was the correct one DNA also. Then the DNA microarray is used for studying the hybridization, comparative genomic hybridization. Then another kind of microarray is the protein microarrays. If we want to identify the interaction of protein with the protein, this is just one example. If we can also use the interaction of the protein with the DNA or with other ligand molecule. Then the tissue microarrays, if we want to, or if we are performing the microarray experiment on the molecules which are present in a particular condition in a tissue. Cellular microarray, which is also known as the transfection microarray, when the microarray is performed on the cellular components. Then chemical compound microarrays, if we are using the chemical compounds. Then antibody microarrays, which is included in the proteomics. And the carbohydrate microarrays, which is also known as the glycoarrays. Glyco is from carbohydrate. So these are the different kinds of microarrays. In future, our focus will be on studying the DNA microarray. What is meant by DNA microarray? DNA microarray are solid supports, and these solid supports are usually of glass or silicon. So the chip that we use as a solid support, it is made up of glass or silicon, and upon this chip DNA is attached in an organized grid fashion in the form of rows and columns. They are at equal distance, as you can see in this diagram. All the DNA molecules, they are immobilized or attached on this solid surface in an organized fashion. Each spot of DNA will be known as a probe, and this probe or each spot of this DNA, it actually represents a gene. This is a one spot, we will call it as a probe, and this probe is a single gene. So this miniaturized technology, it can contain thousands of the gene on a single chip. There are thousands of the gene on this chip. The DNA microarray has several synonyms, like we also call it as the DNA chip. It is also known as the gene chip. It is also known as the DNA arrays or gene arrays, and this is another term which is biochip, which is also used for the DNA microarray. So first of all, we have said that DNA microarray involves a glass or plastic or silicon slide, and then each spot on the slide, it has a different DNA sequence. As you can see in this spot, this is the sequence of the DNA, which we will call it as a probe, and one probe is for one gene. So microarrays usually have thousands of the DNA spots. So thousands of the genes are present together and in an organized fashion on a single glass plate. And DNA microarray is mostly used to find out the gene expression for the expression analysis. And when we talk about the expression, it means we want to study about the status of the protein that we can get from the messenger RNA. So actually microarrays are used to determine the types and quantities of messenger RNAs transcribed. What kind of messenger RNAs? Type means kinds of the genes which are showing their expression in a particular condition, at what type they are showing their expression. We know that all the cells have same kind of DNA. This DNA is same in all the cells. Then the question arises that how do cells that have the same DNA or genes, they end up having different structures and function. For example, some make the part of the cell membrane, some make the part of some kind of other tissues. This happens due to differential gene expression. There is a term which is known as differential gene expression. It means different genes, they are turned on in different cells, and these different genes then determine they are going to develop into what kind of cell. So different genes, they make different kinds of messenger RNAs, and they are making the different kind of tissues or cell. An example of this differential gene expression that we can take is that the bone marrow stem cells, they can differentiate into specialized kind of blood cells because different genes are expressing during the developmental stage. Principle of DNA microarray, the principle is on the hybridization. It is based upon hybridization between the nucleotide. Which nucleotide? We have immobilized the nucleotide on the solid support, and then we are also going to apply the certain samples on it, and there will be the hybridization or attachment of the two DNA molecules together. As we know that the DNA is made up of nucleotide, therefore we are saying that it is based upon the principle of hybridization. Using this technology, the presence of one genomic or cDNA sequence in 100,000 or more sequences can be screened in a single hybridization reaction. What is meant by this sentence? It means that we have various kind of spores, various thousands, hundreds or thousands of spores of the genes in it, and we have a sample, and we are interested in this gene that we are using in our sample, and if we want to detect either the genes which were immobilized on the chip, they contain the gene complementary to this one or not. So these all the DNA fragments which we have immobilized on it, they are single stranded, and we are using the probe also, which is a single stranded. So when we mix it, or when we apply this one over here, then suppose this red spot, it combined with this blue one. So we have identified or screened a single gene from 100,000 of sequences, and their combination occur due to hydrogen bonding that occur between two strands of the DNA. We know that C and G, they make 3 hydrogen bonds, and A and T, they make 2 hydrogen bonds. So the property of complementary nucleic acid sequence is to specifically pair with each other by forming the hydrogen bond between the complementary nucleotide base pair. So this is the principle of DNA microarray. We have to perform the experiment of DNA microarray. Then there are certain requirements for designing of the system, and these include we need a DNA chip. Then we need a target sample, and detection is based due to the labeling of the target sample, and we fluorescently label the target sample. Then we use the enzymes and fluorescent dyes enzymes which can be used for different purposes. Now we will take an example over here also. Then fluorescent dyes, then probes and scanner. So for designing a DNA microarray system, we need to prepare first of all the sample, and if we have to do the relative comparison of the two samples, like we want to compare the patient with the healthy one, then we will do the same for both of these samples. First we will take the samples, and then we have to isolate the messenger RNA. We will add all the required chemicals in it, or we will use the kit in which the two phases will be formed. In the phenolic phase, there will be the amount of the proteins and DNA which were present in the cell, and the aqueous phase will contain the messenger RNA. We will carefully isolate the aqueous phase from this tube. As you can see in this tube, this is the aqueous phase and this is the phenolic phase. We will use the aqueous phase and we will discard the phenolic phase. As we cannot use messenger RNA as such in the experiment, there is a need to convert this messenger RNA into cDNA, and for that purpose we use the enzyme reverse transcriptase, and we will carry out this reaction by using the amino allyl nucleotides, so that these nucleotides can make the cDNA molecule. So we will synthesize the cDNA molecule, because reverse transcriptase enzyme, it copy the DNA messenger RNA molecule into the cDNA molecule, and here we will use the thiamine, not the uracil for this. Then we will use the scissors or the enzymes that will separate the cDNA from the RNA, and now we will save the cDNA and we will discard the messenger RNA molecule. Then the next step is the labeling. Now there is a need to label the cDNA from the different two kinds of cell, and we label these with two different colors. Like here you can see that we have the Cy5 dye and Cy3 dye. Cy5 dye will give the red color, and Cy3 dye that we will label, it will be of green fluorescence. So we will label the two cDNA molecule. This is the unlabeled and this is the labeled one. As you can see, its boundary which shows that it is now labeled. So we will label the molecule. Then we have a chip. We have a chip on which there are spots, thousands of spots of the DNA molecule, which are well mounted. It is a well mounted microarray which has been pre-printed with genes that we will study in this experiment. And now comes the stage of hybridization, that now we will mix both the samples, labeled sample which are labeled with the green fluorescent dye Cy3 and Cy5. We will mix the labeled cDNAs from two kinds of cells together. Then we will place the labeled cDNA strips into a hybridization solution in the center of the room. Then we will soak this microarray chip in the mixture of the labeled cDNA. And then what will happen? The cDNAs that are complementary to the sequences on the microarray, they will hybridize or bind to them. Then we will take out the cDNA chip. We will wash the microarray chip to remove the cDNA strips that are not hybridized to any spot, and then we will move toward the scanner. We will keep it in the machine, and there are filter and laser lights that will detect the presence of the dyes. Fluorescence will be produced if there is binding of the cDNAs which were labeled to the genes which were immobilized on this strip. Then the normalization of the data will be carried out, and then the analysis will be done. The visualization of the microarray results is done using the laser light that causes the fluorescent labels to emit the light. For example, there are different kinds of lasers, like laser 1 causes the red labels to emit the red light, and laser 2 will causes the green labels to emit the green light. Then we take both the images, then we can superimpose or merge the red and green scanned images using the computer, which is known as the normalization. Then we will analyze the data, that if there are red or green same spots, or there are different kind of spots, that I will tell you on the next slide. So this is the overview of the microarray experiment with all the steps that are performed. Like first of all, DNA microarray is prepared by selecting and printing the known sequences of DNA on a chip. So we have taken a chip on which we have immobilized the known DNA sequences, which are about 20 to 70 bases long. So this is our chip, and you are watching these these small spots in which the DNA sequence is immobilized. So then we need to obtain the types of the cell. We take two types of the cell, we isolate their messenger RNA, we prepare the cDNA, and then we label the cDNA with red dye and green dye. After that, we mix the cDNAs together, and then we take the chip and we put these one on the chip and allow them to hybridize. Now the labeled cDNAs, they will hybridize to the complementary sequences on the chip. As we in this example, we are considering the four wells, and in these four wells you can see what is happening. Different conditions are given, like there are four possibilities that only cDNAs with red label they can hybridize, then only cDNAs with the green label they can hybridize. It is also a possibility that in some gene both the green and red label cDNAs can hybridize, and then there is possibility that there is no cDNA to be hybridized. We will allow the reaction of hybridization to occur. After that, the unhybridized cDNAs, they are washed away. As you can see, these are the unhybridized cDNAs, they were washed away, and then comes the function of the laser light. That laser lights are provided which cause the labels to emit the red or green light, and these emissions are captured onto the computer. So we have these four possibilities according to which as you can see that if we provide the red laser light, then what will happen in this case, the red labeled cDNA has bound to this, if we call it as the 1, 2, 3 and 4 well. Then you can see the red labeled probe has bound to the first and third well. So first and third well will emit the red light, while the second and fourth will not emit the light. And then if you see one, when we have provided the laser 2 with the green light, that the green labeled cDNA has bound to the probe 2 and 3, or well 2 and 3. So we can say that the well 2 and well 3, they will emit the green light, and there will be no green light from the well 1 and 4. So we take these images, then these images are merged, and then sports with both labels, we check what will happen to this third well, in which the laser light red has also shown or emit the radiation, and laser light green has also emitted the radiation. So when both the labeled molecules interact with the DNA molecule in a single well, then these spots will appear yellow. And there are certain spots, like in the 4, in which there is no hybridization of the red labeled cDNA or green labeled cDNA, then in this case the spot will appear black. So here you can see different kinds of the spots. Red spot which show that the red labeled cDNA has bound, green spot which shows that only green labeled cDNA has bound, black spot which shows that no binding of any labeled cDNA molecule, and this yellow spot which shows that the both red and green cDNA has bound to this spot. So red only, green only, yellow will be for the both only, and none. And then we will analyze the results. We identify the genes with which the messenger RNAs has shown the expression. There are different kinds of DNA chips which are used in biochemistry and molecular biology. Mostly they are cDNA based chips or the oligonucleotide chip. So cDNA based chip, this kind of chips, they are prepared by using the cDNA. Therefore it is known as the cDNA chip or cDNA microarray or probe DNA. In this case, the cDNAs are amplified by using the PCR, and then these cDNAs are immobilized on the solid support, which is either made up of the nylon filter of the glass slide, which has a size of 1 into 3 inches. Then the probe DNA that is labeled, they are loaded into a spotting spin by capillary reaction. We provide the small volume of this DNA preparation which is spotted on solid surface. It makes the physical contact with these two probes, and DNA is delivered mechanically or in a robotic manner. When one kind of DNA spotting is done, the pin is washed and loaded with fresh DNA to start the second cycle. So this is the methodology with the help of which we are preparing the cDNA chip. Like we load with the help of the spotting spin, which by using the capillary reaction we apply the small volume of the DNA in a small at the small space on the solid surface. When it is spotted, then we wash the pin and load the fresh DNA to start the second cycle. Second kind of DNA chips which are most commonly employed, these are the oligonucleotide microarrays. In this case, oligonucleotides are the short sequences which are designed to match the part of the sequences of known or predicted open reading frame. These oligonucleotides, these probes are often used in spotted microarrays, but the term oligonucleotide array also represents the same, and it also refers to a specific technique of manufacturing. So these oligonucleotide arrays, they are produced by printing short oligonucleotide sequences which are designed to represent a single gene by synthesizing this sequence directly onto the array surface instead of depositing the intact sequence. So this is the difference of oligonucleotide and cDNA. In case of cDNA, we have the intact cDNA molecule, and we amplify the cDNA molecule by PCR, and then each cDNA molecule is immobilized on the solid surface as such, as we have studied on the previous slide. But in case of oligonucleotide, what will happen that these small size oligonucleotides, they are synthesized on the chip, and we add one by one each nucleotide. Like first A is synthesized, then will come the T and C. And this method is known as the photolithography, and we will study about it. Here is the construction of a DNA chip which is attached to oligonucleotide sequences, and here two cycles are shown. So in this case, these are the protecting groups which are provided on the chip. This is the glass plate or glass slide or chip onto which these are the protecting groups. We will mask the protecting groups at the positions in which we do not want to add the nucleotide. Then we will provide certain radiation which will causes the removal of the unmasked group, and there you can see unmasked groups will form. Then we will add the solution of the adenine, and what will happen, the adenine will be added over here along with the protecting group. After that, irradiation is performed at some other place, and all the other remaining places, they are masked, and they will causes the removal of these groups. And now we want to add the cytosine at this position, so we will add the cytosine solution, and it will become bound to this position. As you can see here, it is also irradiated at this position, or this one, so it will form the AC. So in this way, we synthesized directly oligonucleotides onto the glass slide. This is known as photolithography. This is the technique which is used for the preparation of the DNA chip. As you can see, this is the solid surface on which 1, 2, 3, 4 wells are shown here, and this is the desired sequence, which is the desired one. So what will happen, we will add the solution containing the activated A, and this is the solution of activated A, you can see, and we have provide an opaque screen over the spots 2 and 4, because these A's are not required to be added on the spot 2 and 4. This is the spot 1, this is spot 2, this is spot 3, and this is spot 4. So we do not want A to be added to the 2 and 4. As you can see, the first nucleotide is A in 1 and 3. So we will add an opaque screen at these two spots, and we will add the activated A solution. A will get added on it. Then after that, you can see there is a G on the 2 and 4 spot. Now we will add the opaque screen, and we will add the G. So G will get attached to it. Then after, we will add the C. C will be added to only this one. We will mask with the opaque screen over the spot 1, 2 and 3. We will add the C solution, so it will become C, and there will be many more cycles till we get these sequences at the desired positions. This is the comparison of oligonucleotide microarray and cDNA microarray. Like in case of oligonucleotide microarray, you have taken the cells of person 1 or of a condition 1, you have isolated the RNA, you have taken out the messenger RNA, And with the help of the reverse transcriptase enzyme, you have converted the messenger RNA into cDNA, and it is the labeled one, that it will provide the green fluorescent target. It will be hybridized to the microarray, having the short single-stranded DNA which span the entire genome, and then we identify in a single person that what kind of genes are showing their expression. In case of cDNA microarray, we are actually doing the comparison of the normal cells and cancer cells as we have discussed before. We isolate the messenger RNA, we label the cDNA. After making the cDNA from messenger RNA, we combine them and hybridize to the microarray, and then we get different kind of spots, and you know what is meant by each spot. Applications of DNA microarray, we have said that it is involved in gene expression profiling, that in different cells and tissues during the course of development under different environmental or chemical stimulus, and as well as we can also see the gene expression profiling in disease state versus the healthy state. Then it is also used for molecular diagnosis, like we want to identify the diseases, and then for the drug development, if for the identification of new targets, as well as it is also used in pharmacogenomics for the formation of the individualized medicines or the custom medicines. It is also being employed for the comparative genomic hybridization, like we want to assess or compare the genomic content in different cells or in different organisms which are closely related to one another. It is also used for the single nucleotide polymorphism detection among alleles within or between populations. Then it is also used in CHIP studies. CHIP is a technique which is the chromatin immunoprecipitation, in which we determine the protein binding site occupancy throughout the genome, that at what position the protein bind to the DNA. This is a technique of DNA protein interaction identification, and it make use of the chip-on-chip technology. The main advantages of DNA microarray are it provides the data for thousands of the genes, that we can get the data of thousands of genes together, and we can carry out experiment for these thousands of genes in one experiment instead of carrying out many experiments. It is fast and easy to obtain, and it is huge step closer to discover the cures for the diseases and for the cancer. And in this DNA microarray, we can use the different parts of the DNA to study the gene expression. The main disadvantage of DNA microarray is that it is too expensive to create, to prepare the chip. It is not economical, and the production of too many results at a time, it requires longer time for the analysis, which is quite complex in nature. Even when we talk about the meta-analysis, in which I told you that we take data from the databases, there are various kinds of microarray databases. Once an experiment is performed, a large or huge amount of the data is obtained. That data is then submitted to the particular microarray data bank, and then it can be retrieved, and then different kinds of analysis can be performed, and it takes a long time for the analysis. That's why it is complex in nature. Another drawback is that DNA chips do not have very long half-life. So whenever it is being synthesized, then it is good to be used as you get the DNA chip. So this was all about today's lecture, in which we have studied about an advanced technique of DNA microarray, that we are performing various kinds of reactions together on a simple chip, that thousands of millions of gene expression can be detected by using the cDNA on a single microarray chip. We have studied about the requirements and the principle of DNA microarray, also discussed about the types of the DNA microarrays, its as well as its applications, and then we have also studied the advantages and disadvantages of DNA microarrays. This was all about today's lecture. If you have any question, please feel free to ask. Thank you.